The double helix, discovery of DNA's structure. At the beginning of the 20th century, scientists already knew that hereditary traits, such as eye color or the shape of a pea pod, were passed from one generation to the next. But how exactly this happened remained a mystery. No one knew how genetic information was stored and transmitted. Physicists and chemists had already uncovered the secrets of atoms, but the secret of life, especially heredity, was still elusive. Everyone believed that a molecule must lie at the heart of the process, one that carries this vital information. There were two contenders: proteins, with their complex structure and diverse functions, and DNA, simpler, and at first glance, far less interesting. Most scientists bet on proteins. However, in 1944, American scientist Oswald Avery demonstrated that DNA is the carrier of hereditary information. Unfortunately, his discovery was largely ignored at the time. Then, in 1951, two young scientists met at the Cavendish Laboratory in Cambridge and would change history. James Watson, a 23-year-old American, and Francis Crick, a British physicist returning to science after World War II. Yes, that's right. Francis Crick decided to switch from physics to biology, driven by his passion for understanding life. They became instant collaborators. For them, science was an adventure, but also a race. They feared someone else might discover the structure of DNA before them. Their approach was unconventional. Instead of relying on lengthy experiments, they worked through theoretical models, constant discussions, and wild hypotheses. They were convinced that if they could uncover the shape of DNA, they could unlock the mechanism of heredity. But they weren't alone. At King's College in London, another team, Maurice Wilkins and the brilliant Rosalind Franklin, was working on the same problem. Franklin was an exceptionally skilled X-ray crystallographer who had produced the clearest images of DNA to date using X-ray diffraction. The problem? Franklin believed she was leading the project. Wilkins thought the same. Tensions rose, and eventually they worked separately. Meanwhile, Watson and Crick used some of Franklin's data, without her permission, to build their models. Their first model was incorrect. They had placed the sugar phosphate backbones on the inside and the bases on the outside. Franklin immediately spotted the error and criticized them publicly. The result? Embarrassment, and an official order from Cavendish leadership forbade them from continuing work on DNA. But failure didn't stop them. Francis Crick would later say, "To arrive at the right answer, you have to go through mistakes." Throughout 1952, Watson and Crick continued to read, discuss, and analyze all available data. At the same time, world-renowned chemist Linus Pauling was closing in on DNA as well. The pressure was on. Then came the breakthrough. They gained access to photograph 51, taken by Rosalind Franklin. It clearly showed that DNA had a helical structure. With that image, Crick's knowledge of symmetry, and Watson's understanding of base pairing, they built the model we know today, the double helix. In 1953, they published their revolutionary theory: two sugar phosphate chains twisted into a spiral, with nitrogenous bases paired in the center, adenine with thymine, guanine with cytosine. This structure explained not only the stability of genetic information but also its ability to replicate, the key to heredity. In 1962, Watson, Crick, and Wilkins received the Nobel Prize for their discovery. Rosalind Franklin did not. She had died of cancer in 1958 at the age of 37. The Nobel Prize is not awarded posthumously,